Hello. First of all, thank you all for being here and for joining me today. So the seed is a major innovation of land plants and constitutes a turning point in its evolution. The diversity of seed is fascinating, as you can see here, just a sampling of many and different seeds. So to approach the understanding of these enigmatic and diverse structures, we wanted to get an insight into the genes that are involved in its development. This research project has been focused on 5 different species, and today I'm going to talk to you mainly about ginkgo and nidum in order to have a better idea of the possible origin and evolution of the seed from a molecular approach. All right, so around 400 million years ago, there were no seeds, but reproductive structures of these seedless plants, like lycophytes and ferns, had some features key for the subsequent evolution of the seed, like heterospory, which is the production of 2 different kinds of spores, so the megasporangia that give rise to the female gametes, and the microsporangia that produces or gives rise to male gametes. Another key feature is the endosporic development, which is the development of the gametophyte inside the spore. And later on, the integument evolved, which ultimately is the key novelty in the evolution of the seed, as this structure is the one that ensures the protection of the embryo and tolerance for desiccation for very long periods of time. However, the evolution of the integument, and in fact of the seed, is not very clear yet. In fact, there are 3 different hypotheses that aim to explain how the seed and the integument itself evolved that are still under discussion. So based on the aerial of Taxus, the de novo hypothesis suggests that the integument evolved as a new structure, the aerial here popping out. Then the telome origin hypothesis. So this is the de novo hypothesis represented. Then the telome origin hypothesis is based mainly on fossil record, and it suggests that the integuments evolved by fusion of telomes around a single fertile sporangium. Here we have represented the sporangium with this gray structure. Then the synangial origin hypothesis is based mainly on living lineages of ferns and the pollen cones of cycads, for instance. And this hypothesis suggests that the integuments evolved by sterilization and fusion of sporangia, leaving only one fertile sporangium in the center. The fossil record may also support this hypothesis, as these telomes may have been fertile at some point. So these 2 hypotheses are not necessarily mutually exclusive. So, but in fact, the seed is the result of fertilization of the ovule. And here we have represented 2 different types of ovules, one of gymnosperms and one for angiosperms. And the ovule is defined as a megasporangium or nucellus that is covered by the integuments, here represented in yellow. So most gymnosperms have one integument covering that nucellus, and the opening that allows the pollen to enter and fertilize the megaspore mother cell, the micropyle, is located opposite to the funiculus or the stalk of the ovule. Then most angiosperms have 2 integuments, inner and outer integument, and the opening and the micropyle, which is the opening that allows the pollen to enter, is located next to the funiculus of the ovule. This is due to an asymmetric growth of the outer integument. So again, this is for most species. However, there are a lot of variation and there is a lot of diversity of seeds. And in gymnosperms, which is our focus, there are several gymnosperm ovules that have structures that are associated to these ovules. So for instance, in Taxus, there is this aerial that develops at the base of the seed. And as the seed matures, the areoles also grows and covers the entire seed, and it becomes red and fleshy. Then in ginkgo, there is also a vegetative structure at the base of the ovules. This structure is called the collar. And yeah, it remains at the base of the ovules of ginkgo. It's unique to ginkgo. In Nethalis, in fact, there are several structures or there is a structure that covers always the seed, but it can vary in numbers as well as in the morphology. Some Nethalis have these structures fleshy, whereas in some species of ephedra, for instance, these envelopes or these extra structures that cover the seed are completely dry. So the homology of all of this structure has been a matter of controversy, and it's very difficult to assess the homology of a structure, but it's been a very intriguing and a debated issue in plant morphology. So many people actually consider these extra envelopes of Nethalis as extra integuments. So in order to give this homology, this discussion, like a different perspective, what we did is that we started looking at the molecular mechanisms and which genes may be involved in the development of the different structures of the seeds in these species. So we started by what is known from, or the genes that are known to be involved in ovule and seed development. And what we know the most is from the model species Arabidopsis thaliana. So here we have a scanning electron photograph of the Arabidopsis ovules. And this is a drawing of one of those ovules with the two integuments here. And I'm going to use this to illustrate with different colors where each of the genes has a function. So for instance, the gene A integumenta is involved in the proper development of both integuments. And at the bottom, we have the mutant, so A integumenta mutant. So when this gene is not present, the ovule doesn't develop integuments. BEL1 is also involved in the proper development of both integuments. When this gene is not present, there are no integuments and there is a fleshy structure at the base of the ovules. Inner no outer is a gene that is specific to the outer integument. When this gene is not present, there is only one inner integument developing. Then aberrant testa shape, which is a canary gene, is also involved in the proper separation of both integuments, and this is due to its role in the polarity or that flat identity that have the integuments. So once the integuments have been initiated, they have to elongate to cover the nucellus, and there are several genes involved in this process as well, like short integuments 1 or unicorn, which is super cute and it has a very cute name, and it's involved in the proper polarity of the outer integument, and the mutant has these outer growths coming out of the outer integument. So the story gets a little bit more complicated because each of these genes belongs to a different gene family. So to assess the evolution of these genes is going to be very difficult. And in fact, each of them has a unique evolutionary history as they are members of different gene families. So what we did is that we started looking at these genes in ginkgo and niru. And we also looked at a gene that is called wuschel that I didn't mention before, but wuschel is expressed only in the nucellus, but it's involved also in integument and development. So wuschel in ginkgo, we were able to see that is expressed. This is by in situ hybridization, which is a technique that allows you to see exactly where and when the gene is expressed. So where you see expression is when you see this blue or purplish color signal in the tissue. So we saw expression throughout the ovule, as well as in the pollen grains and in the leaves. This is for wuschel. A integumenta, so I'm going to leave you here on the left a cartoon that shows the expression of those genes in Arabidopsis, because I know there are too many genes, too many things to keep track of. So I'm going to leave a cartoon there so that you can compare with the results that I'm going to show you right away. So in ginkgo, we saw that I tegumenta is expressed only in these cell layers at the base of the ovule, which in fact are called the abscission zone, because as the region through which, when the seed matures, it's going to fall off through that region. So this is the abscission zone. Then is expressed also in the pollen chamber. The pollen chamber is the tip of the nucellus. When the pollen enters the ovule, it arrives to the pollen chamber, and this chamber kind of like degenerates, bringing the pollen to the egg cell. And in fact, we saw expression also in the megaspore mother cell, as well as in the pollen grains and the leaves. All right, in Nidum, we saw expression of tegumenta in the nucellus as well as in the integuments. Then Bell, which is also expressed, it also functions in integument development in Arabidopsis. We saw expression very similar to Arabidopsis, to integumenta, I'm sorry. And we saw expression at the base of the ovules in the abscission zone, the pollen chamber, and the megaspore mother cell. For Nidum Bell, we saw expression in the nucellus, highly expressed when the seed is mature. Canary, remember that canary genes, I talked about avar and testa shape, which is involved in that polarity of the integuments. And in ginkgo, we saw that is expressed in the nucellus throughout the ovule later on, as well as in the pollen grains and the leaves. For Nidum, there are two copies of canaris. So one is expressed in the nucellus and the megaspore mother cell, and the other one is specifically expressed to the megaspore mother cell and the tip of the integument. Unicorn, also a polarity gene, mostly for the outer integument. We saw in ginkgo that is expressed at the tip of the ovule, as well as in the pollen grains. There are also two copies in Nidum. One is highly expressed in the nucellus, and the other one is specific to the megaspore mother cell and the tip of the integuments. All right, so with this data, we can start making some conclusions. So the first one is that the evolutionary history of these genes is very complex, as you may imagine. And here I represented the different structures, the seeds of different species, Arabidopsis, and the two species that we worked on, in order to have like a visual representation of the expression patterns that we detected. And as you go through, you're going to see that it's very difficult for each of these genes to make conclusions on whether or not their function is conserved. But this is also because we're comparing species that are morphologically very different. So this is not helping us a lot on making conclusions on the evolutionary history of these genes. Because if we look at the structures of these species, so for instance, here on the left, we have a drawing of the three different tissues in Arabidopsis ovules from the proximal distal perspective. So we have the funiculus, then the chalazal region. This is the region from which the integuments develop, and then the nucellus. In ginkgo, it's more difficult to understand this structure. So we have the funiculus, or the stalk of the ovule, and then there is a region on which the nucellus and the integuments are completely fused. And this region is very difficult to separate the two tissues anatomically or morphologically. We don't see much difference between these two regions. And this whole thing, this whole part of the ovule is called the pachychalaza. Then the integuments become free from the nucellus, so they are not fused, and there we can see the difference between the structures. So we know from Arabidopsis that wuschel is a nucellus, it's a meristematic gene that is only expressed in the nucellus. And then Bell is a chalazal gene that represses wuschel to the nucellus. And then we start seeing something in ginkgo. For instance, that wuschel is expressed only in the pachychalaza, or is very strongly expressed in the pachychalaza. So giving this structure a meristematic identity, because wuschel is a meristem gene. And then we were able to see that Bell may be at the base of the ovule. So probably the functions or the main role of these genes may be conserved, but it's just difficult to reconcile due to these morphological differences. But when we start understanding the identity of these tissues, we start seeing that the main role of these genes may be conserved. Something that was very clear is that we know that these genes are integument genes in Arabidopsis. And we saw all of them expressed in the sporangial tissue. So we saw expression in the megaspore mother cell as well as in the microsporangia. So the pollen grains, we also saw expression of all these genes. And compared to previous works, we saw that some of these genes that have been studied in lycophytes, ferns, and mosses, they're also involved in sporangial development. So this suggests that the integument developmental network was co-opted from a sporangial developmental network. And this allowed us to give for the first time evidence showing that, molecular evidence showing that the integuments may be the result of sterilization and fusion of sporangia. So this kind of supports this hypothesis for the evolution of the integuments. Then a conclusion specific to Natalis, or to Nirum in this case, is that in fact we looked at genes that are known to be involved in integument development in angiosperms, and we didn't see expression of any of these genes in the outer envelope, so those extra envelopes that have been many times considered as integuments. So at least we know that there is no expression of those integument developmental genes in these structures. However, many more studies are still required, probably doing transcriptome analysis on the different tissues of these species would be good in order to have a more clear panorama or vision of which genes are involved in the development of these structures in order to start giving them like a molecular identity, let's say. And in fact, that's what we did for many of the species. We started doing transcriptomic analysis. In Ginkgo biloba, we dissected the young ovules, the integuments, the megagametophile, the collar, the pollen cones, and the leaf. And we sequenced separately all of these structures in order to have a better idea of what are the genes that are involved in the development of the other seed structures. Because with the approach that I just showed you, which is a gene candidate approach, is very targeted. So we look at specific genes that we already know from Arabidopsis. But if we want to know something different, or maybe something that is unique to Ginkgo, we have to start sequencing and understanding which are the genes that are expressing these tissues. So that's what we did. And we also did it for 2 species of Natalis, 2 ephedras with different morphologies in their ovules. So we dissected the young cones and we sequenced that part, as well as the shoots, the pollen cones, the ovules, and the bracts, which are those envelopes covering the seed. In this species of Ephedra californica, we have, or the species has dry envelopes covering the ovules, and Ephedra antisyphilitica has these fleshy envelopes or bracts that I mentioned here, covering completely the ovule. What we saw is that we confirmed through this transcriptomic analysis to see the differential expressed genes. There are several genes, very well-known gene families that are involved in the development of these structures. However, we did not see any of those integument developmental genes highly expressed in these structures, which corroborates our gene candidate approach. And we were able to detect some unique genes in each of these species that we're still characterizing. So in order to start like our conclusion, it's important to highlight that these structures are very unique throughout all seed plants. They are anatomically very diverse. That is what I'm representing here. The integuments, you can see that the vascular traces in each of the species are unique. So that makes assessing the homology of the seed much more difficult. There are many different structures associated, and this is only in gymnosperms. In angiosperms, there is also a whole unique diversity of seed morphologies. And we see that also in the morphology of the seeds, as I show you at the beginning. And another thing that adds up to the complexity on assessing the homology of these structures is the evolutionary history of seed plants, because there are many lineages, extinct lineages. So we're working on the species that exist today, and given that complexity in the understanding of the evolutionary history of these species, it has been very difficult to assess the homology of these structures. However, if we start combining these molecular approaches with morphological approaches and anatomical as well, as I showed you before, it allows to start to understand better the identity of each of these structures, and probably will allow us to understand better the evolutionary history of these species and these structures. And this will be done only if we start looking at all of these different species from a more careful perspective, and without necessarily using what we know from one model species as reference to describe and understand this species. We have to start taking a more diversity-based and a non-model species approach in order to start understanding better these structures. All right, so something else that is very interesting is that even within angiosperms, there must be many differences in the regulatory networks of these species. So for instance, the model species Arabidopsis, here we have the carpel illustrated with this orange structure at the center of the flower, and the placental tissue, so the meristematic tissue that gives rise to the ovules, that meristematic activity gets turned on in the inner layer of the carpel once the carpel is already established. So, however, in other species like Petunia, that meristematic activity that gives rise to the ovules, it's always on. So that suggests mainly differences in the gene regulatory networks that control the initial establishment of ovule development. And if we're seeing differences within angiosperms that we could say have more similar structures, imagine what we see when we start looking at other species like gymnosperms with very different ovule morphologies. So definitely more studies in gymnosperms are still required, and different type of studies in order to better understand other characteristics of this regulatory network. But with our results so far, we were able to show that molecular evidence that suggests that the integuments and the seed may have evolved by the synangial origin hypothesis, suggesting that the sporangia sterilized and fused, leaving only one fertile sporangium in the center. And with this, I want to conclude showing this diversity and the importance of studying different species. As you probably saw from this talk, it's very difficult to draw conclusions based on what we know from one specific species in relation to this enormous diversity of structures that we find. And with that, I want to thank all our funders, my home institutions, and yes, thank you all of you for joining me, for being here today, for tuning in today. And I'm happy to take questions, and you can email me or contact me anytime. I'm always happy to talk about seeds and plants.